All right. Today's lecture, we're doing the generalized animal cell. And let me call up the screen here so that I can begin the lecture. All right. Can you guys see the chapter 3, part A, cells, living units? If you can see that, please type yes in the chat box real quick. And I'll have a look. Okay, good. You guys can see that. Excellent. Now, today's lab follows this lecture at 10:30. Now, I do realize a lot of you, we're going to be ending lecture at 10. I too have to drive across town to get to to get to the lab. So not too far across town, but it's still a drive. And I understand if you're going to get there a little late, but get there as you can, and I will too. So if I'm not there precisely at 10:30, don't leave. Okay, those of you that do arrive on time and are getting there, that's okay. We'll all be real patient with each other with regard to this. All right, so we begin with the cells. And a cell is the structural and functional unit of life. How well the entire organism functions depends on individual and combined activities of all of its cells. Now, a cell essentially is a living entity. It's the basic unit by which your body is able to replicate and to carry on its functions. It has organelles, which are multiple organs on the inside, and it's surrounded by a boundary, the cell membrane. This cell membrane is composed of molecules called phospholipids, which we are going to get into here. Different types of cells represented up on the screen. We see neurons, we see skeletal muscle, we see smooth muscle, epithelial tissue, and on to the specialized cells of sperm, which only males create, and they only possess half the genetic material that all the other cells in the body have. And we'll get more to that when we get to reproduction and development. So the plasma membrane is the flexible outer boundary. The cytoplasm is the intracellular fluid which has and contains the organelles. And then we have the nucleus, which is got the DNA. It contains all of your programming within there. And again, remember the big difference I had mentioned before about cells was that prokaryotes carry their genetic material in a plasmid. Eukaryotes, which we're going to talk about exclusively today, contain their DNA within the nucleus. That being the big difference. Big picture here showing us a cell. And we're going to go over all the different parts that we see here. First and most notably is the external boundary, the cell membrane. And then the second big feature that you will notice, especially when we're doing microscopy work, is the nucleus itself. And there's going to be a nugget on the inside called the nucleolus. All of these organelles have function with how this cell carries out its life and its purpose. And we're going to go through them today. All right, first thing are the fluids: extracellular fluid, interstitial fluid, intracellular fluid, all base composed of water. Very important to keep well hydrated. We have fluid that surrounds the cell, the extracellular fluid. Interstitial fluid is the fluid that's found in between each individual cell as they stand side by side. Intracellular fluid, of course, is all of the liquid on the inside of the cell. All right. Membrane lipids form what we call a lipid bilayer. Now, bilayer means that there are two layers, and we'll show you just what we mean. There are proteins that float around through this, and there are surface carbohydrates, surface sugars that form something called the glycocalyx. Now, this glycocalyx, make a note, is a basis for immunological recognition. Your body's immune system uses this sugar coating to help identify self. This is the basis of when your body rejects tissue that doesn't belong, like when they talk about tissue rejection that comes from a transplant or a graft. The body doesn't recognize this glycocalyx that comes in with the other tissue and works to get rid of it. So it's the basis of this structure. And we'll show you more here. All right, we have, let me get to a better view on our slideshow. We have here up on the screen the plasma membrane, and I'll enlarge it just a bit. And here is our lipid bilayer. These individual molecules are the phospholipid molecules. These large ones here are the proteins, and they float amongst these phospholipids. We have two layers of phospholipids, an outer layer here, and we have an inner layer right here. The phospholipids themselves, you notice, have a circular head, and then they have these two fatty acid tails that we see. Now, these fatty acid tails, if you look closely at the diagram here, you will note they are not perfectly straight. You will note that they have a little wiggle to them. They have a kink. And that kink is just part of the characteristic of the chemical bonds that make up this fatty acid. But by having this kink, it prevents the phospholipid molecules from lining up too close to one another and producing a rigid structure. No, this kink helps to confer fluidity so that these things are able to shift and move around. What you're looking at here looks quite static, but it is not. It is quite dynamic. It is as dynamic as looking at a cluster of plastic, brightly colored balls floating around on the surface of a pool. So in your mind's eye, think of a swimming pool. Most of you know what a bounce house is, with all the little balls bouncing around and you're bouncing around inside of it. So now think of a swimming pool with these brightly plastic balls floating on the surface. And let's say you've got a lot of them and they just cover the surface of the pool. Well, if you throw a small child in the pool and tell them to swim amongst those plastic balls and keep as much of your body underneath, let me just see your head. You would just see like the little head of a child floating amongst all of those plastic balls in the swimming pool. That's what this construct of this diagram shows you. This is how these proteins float around, just like that small child's head within that pool of balls. So these proteins are able to move and be moved by the cell itself. Note here, elements and filaments of the cytoskeleton that are attached to these proteins. The cell is able to move these proteins through its membrane. It moves them in place to function as receptor sites. It moves them to function them as perhaps linkers and joiners, to be adhesive to other cells side by side. So your cell membrane is very dynamic. All of this stuff moves. Now, cholesterol here is the yellow molecules. So where you see the yellow molecules are cholesterol. So cholesterol is vital as a function because it contributes to the fluidity of this phospholipid bilayer membrane. Now, because of the nature of the phospholipid molecule, we say that it is amphipathic. Amphipathic. Where have you heard the term amphi before? Anybody? Type it in the chat box. Let me see. Anybody? Amphibian, someone put. Very good. So that prefix, amphi, all right, is with amphibians. And what can we say about the nature of amphibians? Anybody? They do they live exclusively on dry land? No. Do they live exclusively in the water? No. Amphibians are things like what? Frogs and newts and salamanders. They've got to be around water and they have to be around land. Well, same thing with these phospholipid molecules, the way that they're creating, they have a characteristic of how they react to water. And so we look at these and say, we have a hydrophilic and a hydrophobic layer. If we look here at the diagram, the heads are hydrophilic. They like being around water, so they're going to orient themselves externally towards the water-based extracellular fluid. And they're going to orient internally towards the cytoplasm, which is a watery environment. The fatty acids that we see here on the inside being associated with cholesterol, they don't like being near water, just like normal fats don't mix with water. That's how this is too. So we find all of the fatty acids here. So we say that this is a lipid bilayer, and indeed it is because we have two separate layers of phospholipid molecules, but it has three, count them, three distinctive regions. A hydrophilic external, hydrophilic external, hydrophilic internal, hydrophobic internal on the inside, very inside layer here. So hydrophilic, hydrophobic, hydrophilic. Now, if this is hydrophilic and this region we say is hydrophobic, what can we infer about the protein that's floating within this phospholipid bilayer membrane? We can infer here that this part and this part would also be hydrophilic. This part would be hydrophobic because it matches the characteristic of where it is set. And this is why these protein molecules are able to maintain their orientation in the membrane because, a good question is some people could say, well, what's to keep them from just sort of flipping around, and being unstable? Well, it's that chemical characteristic that helps to keep them secure. Even though they can be moved, they still maintain an orientation. Now, that's not to say that they can be disrupted in their placement. Oh no, there are several drugs, gases that can displace the movement of the proteins in the cell membrane. And one of these you're familiar with if you haven't had personal experience, and that's anesthetic gases. Anesthetic gases disrupt the placement of these membranes, and this is how they can contribute to knocking you out, removing your consciousness, because what happens is these are moving around, and so they're not in the proper place for neurotransmitters to hook up with. And so that's what contributes to your falling asleep. Conversely, when they blow off the gas, you're coming out of it, and it takes degrees. It's not just like you suddenly wake up, you have to recover. And that's why after surgery, you have just this separate area called recovery for the patients, where they're maintained until they have fully come out of the gas. And part of that is reestablishing the placement of these proteins back in. All right, let's see. Amphipathic proteins with channels. Now you notice some of these proteins have channels, like here in this diagram here. So there's a channel. That means things can come in and items can be moved out, all right, of this. This is our glycocalyx, as indicated in the diagram. The hexagonal structures that you see here are sugar groups. Each one is a sugar molecule that we see here. Each one is a glucose molecule. And it looks like a forest of them growing up and out. Note that it's coming and extending off of the protein, and they're also coming off and extending off of the phospholipid molecules. How this is shaped, like where you see the branch points, everybody have different branching points to the glycocalyx on their cells. This is what contributes to you. This is what makes you unique immunohistochemically, is the presence and how these are oriented and placed. And so when they do tissue grafts and transplants, they're looking for a good match of your features here with the donor, which you're going to get it from. And what they're looking for is just a match of four. Four. There's many, many of them, but a good match is considered. Well, if you can match with four of them, then the tissue has a good chance of taking. The transplant has a good chance of taking. And so that's what we call the IHC marker, the immunohistocompatibility marker. It's made up of the glycocalyx, proteins with glycomarkers. I have the word receptor there in the outline to indicate that to a large extent, these proteins, as they float around, again, are used as receptors. Now, this is a leaky feature. This is a leaky structure. Water can pass through here. So the cell has got to be able to maintain its water balance, and it does this through processes called diffusion, osmosis, and filtration. I just mentioned them here. We're not going to get into it in depth because that's more physiology, but these are the processes the cell uses to maintain water balance as things leak across. Top of the second page for the lecture outline, movement across the membrane is an active process. Things are coming and going all the time. We have active transport and under a process called endocytosis. Now in endocytosis, that's the cell taking material in. And so phagocytosis is where the cell is eating, it's consuming something. Penocytosis is where the cell is going to be drinking. It's actually going to be taking up either little amounts of lipid or actively taking in little amounts of water. And then we have receptor-mediated endocytosis. This is very specific, especially with regard to neurochemicals and hormonal control, where we have the binding of hormones and neurochemicals with specific receptors, and the cell is able to bring these molecules in so they can perform other functions within the cell. And these receptor molecules are called clathrin lined, clathrin lined. And I will show you here in a moment what I'm talking about. But clathrin is a it's an anchor atom, an anchor molecule that the cell uses to help bring in a part of itself in the phagocytotic process. Most of the cells that undergo or do phagocytosis in a dramatic way in your body are the white blood cells. They function to phagocytize pathogens, bacteria, fungus, and virus. I mean, it's an active consumption going on even now as you're just sitting there listening. Your immune system is actively engaged in this phagocytic process to keep your immune system, to keep your whole circulatory system sweet and clean and free of any type of infection. Now, organelles, some are made of exclusively membrane, and some are exclusively made of protein. So let's have a look at some of these. Let's see. All right, we talked about the lipid bilayer, glycolipids, cholesterol. Here was just the picture of the molecule itself and the fatty acid tails with the kinks that help to keep the phospholipid layers moving. Now, someone just asked a chat question. Will all of this be on the quiz after lecture? No, no. And remember, there is going to be a lecture. I think I've already opened it up. I already have it there. It should open. It's just a practice quiz, people. It doesn't count for points. It's not extra credit. And it's basically what we talked about Monday. And see what you can remember. I can't, I'll tell you one thing. I don't really remember if I have anything from today's lecture on that quiz or not. It'll be a surprise. But if you don't remember a single thing from the lecture, it's okay. It doesn't count against you. The whole function, the whole point of taking the lecture is to kind of see my style, not to test your knowledge, but for you to see kind of my style of how I've set up the exams. So don't stress about it. Don't worry. Okay? Okay. Let's see, where are we at now? All right, integral proteins, we talked about peripheral proteins. This shows you the different functions. Now, I talked about mostly receptors and linkers, but proteins are also used for enzymatic activity, cell-to-cell recognition, attachment to the cytoskeleton. Here's our linker activity, receptors for signal transduction, all kinds of things proteins are used for. All right. And this just goes through the individual ones, bigger picture. Glycocalyx, I talked about that. All right. Now, junctions. Cells are held together with tight junctions, desmosomes, and gap junctions. These are all characteristics of the cell that help to hold it together. Because if you don't have these, you begin to fall apart and you can develop a really terrible syndrome. And the name of the syndrome, actually the name of the disease, is called pemphigus. And there's several forms of pemphigus. And let me make sure that everybody can see this meeting. Pemphigus. This is a disease where some of these junctions go missing, like the desmosomes for some. And you might think, okay, well, this is just a really small structure or feature. What does it matter if it kind of is like not present or it doesn't work too well? I mean, you can't even see it. How bad can the compromise be? It can be very bad. Pemphigus creates brittle, not brittle skin, but overly delicate skin. I mean, people's skin can just come away if you pull on it. If you were to grab the finger of someone who has pemphigus and their hand looks intact, just like yours, and you were to twist on the skin and pull, just like if you were to do that now, that action would tear their skin off like a sleeve. That's how delicate their skin is, fragile as a butterfly's wing when this occurs. So these people have a very compromised quality of life with regard to skin. Let me see if I have a picture here to show you of the junction itself. Ah, here we go. So these junctions. Now, desmosomes are kind of like these button welds. The tight junctions are here like a big plastic band. Think of a tight junction as looking like, you know, the plastic that's on beverage cans that hold them together in six packs. That's kind of how like the tight junction works for cells. The desmosomes here and here are like buttons in fabric. So imagine like two buttons, a button over here, a button over here, and they're kind of stitched together here, and that helps to stabilize and hold the cells in place. Because of course, this is a cell, this is a cell, and this is a cell. And all of these connectors help it to stabilize itself. Now, gap junctions on the list are the only communicating connections between cells, gap junctions. And let me see if we have a picture of this. Okay, this is a description of the desmosomes, cadherins, which are sort of the buttons. That's what rivet-like means. It's a button-like linker. Ah, here we go. So here we go. Here's like a button. Here's a button. And they're all kind of attached one to another here. So that's a desmosome. Gap junctions we see here communicate. Electrolytes and molecules can pass between cells to communicate. And so all cells have gap junctions, but the tissues that are really rich in gap junctions, of course, are cardiovascular tissue and smooth muscle. Someone asked in the chat box, is that how it gets as we age? No, no, skin ages, but not because we've lacked desmosomes. No, most everyone that's aging, you know, that can age has desmosomes and gap junctions and everything else. People that are born with pemphigus are usually dead by the time they're 30. They don't survive much because there's just too much trauma associated with their condition that eventually they also develop cancer of the skin because of it. So now let's see, cell junctions. Here is the gap junction here and here. There we go. All right, passive transport, etc. ATP, all you have to know, okay, ATP is the energy produced by cells. And a specific organelle produces it. We'll talk about it shortly here. Adenosine triphosphate. Adenosine triphosphate. Let me see if I can type that in. Adenosine. That's ATP. This is the fuel that your body creates. It's the energy. Active transport uses ATP to transport items through and across through your cells and across the cell membrane. Passive transport does not require any ATP, so none will be used. And this just goes through the diffusion, osmosis, it talks about dialysis. I'm not going to overly stress it again, because like I said, it's part of physiology. I just introduce it here to you. Now, someone had asked in the chat box previously about polar and nonpolar. Yes, the phospholipid molecule is considered a polar molecule because it has an orientation with regard to water in this fashion. We have a hydrophilic head and a hydrophobic tail. The hydrophilic head would have a slightly positive charge, whereas the fatty acid tails are going to have a slightly negative charge. And so this is what will create a polarity across the molecule. So to answer that question, yes, polar molecule for the phospholipid molecule. All right, lipid soluble, nonpolar. It has just a description there of the nonpolars. All right. This just shows you diffusion of lipid soluble. Again, this is more physiology, so you don't have to pay too much attention to that. All right. And moving through, you don't have to know anything about aquarins, but it's there. Osmolality and osmolarity, you definitely do not need. So don't stress this. Don't ignore this. That's going to come up when you take physio. But it's introduced here because it does talk about cells. But I will not test you. This will not be testable at all. Tonicity, no, you don't have to bother with that. We will talk about tonicity when we get to red blood cells, but it's not important for you to know right now. So just save that for the red blood cell lecture when we get to cardiovascular. And then, let me see, I have another PowerPoint here. Just give me a moment to pull it up. Okay, as I look for it here, I should be able to browse and see what I'm looking for here. Okay, now I share. Where is it? It's gotta be in here somewhere. So if you have any questions at this point, put them in the chat box and I'll take a quick peek as I locate my other PowerPoint here. Where did it go? I had it, I thought, set up. No, that's not it. Tell you what, let's go back. Oh, no, not that one. All right, let's see. Let me answer a couple questions while I'm looking. Let's see. Someone asked about the book. The edition, 10th or 11th, either one will do. So you can bring and have for yourself either one. Doesn't matter. Again, they haven't changed to human that long. Someone says, do we focus on studying the outline over reading the textbook, or should we be reading a certain number of chapters? There's no assigned chapter reading. What you do is you take your outline and use the PowerPoint and study the content as I go through it just this way, and use your textbook for clarification and for enrichment, because there might be one or two features that I mentioned very quickly and that they're on the PowerPoint, but you should make a better notation of it in the outline. But for testing's sake, everything I have in the outline is testable material. Do we study off the PowerPoints along with the outline? Yes, I just addressed that. So, all right. No,
If we have lab today at 10 or after this, do we meet on campus?
Yes, all labs are on campus.
Okay.
All labs are on campus. All right. Well, I can't find my exclusive PowerPoint, so I'll just use the diagram that I've been using so far here. So let's just go back. New screen share. All right, animal cell lecture. Let's get back to that one. Okay. Now let me see. Where are we at here? Just go back. All right. So I'm just gonna get back up here. We'll just use this. Okay. So going back in the PowerPoint, we're gonna go through the organelles real quick for the rest of the lecture here. All right, the nucleus I've already addressed. Nucleus full of chromatin, DNA with histone proteins. Now make a note, histone proteins are round proteins around which the DNA molecule organizes itself so that it doesn't become tangled up. So it's a good organizer protein, a histone. A nucleolus is at the center. We'll discuss that a little more. The envelope of the nucleus is full of pores. Note here in the diagram up on the screen that you have a lot of pores. And this is for messenger RNA molecules to exit the nucleus out into the cytoplasm so that they can be translated. Transcription, the creation of these messages, occurs within the nucleus. Translation is going to occur by ribosomes out here in the endoplasmic reticulum. We have two types of endoplasmic reticulum. We have smooth and we have rough. The smooth, SER acronym, processes lipids and carbohydrates. You need to know that function. Smooth endoplasmic reticulum is membranous, and we say it is smooth because it does not have ribosomes. There are no ribosomes associated with smooth ER. Rough ER, acronym RER, is associated with ribosomes. And this association with ribosomes, as we see here, diagram up on the screen, Here is our rough endoplasmic reticulum. Lots of ribosomes gives it a rough, pebbly appearance, especially when we're looking at an electron micrograph of the cell. The rough endoplasmic reticulum processes protein. I know that's not there on the outline. I'm wanting you to add it. I'm telling you to add it. The Golgi apparatus that we see here, here's our Golgi. This Golgi apparatus or Golgi complex or body is the UPS. It's the FedEx or the UPS of the cell. It packages and it ships product. It's directly found between the rough endoplasmic reticulum and the cell membrane. It has two surfaces, a cis and a trans surface. The cis surface is closest to the nucleus. It's the receiving. The trans faces the cell membrane, and the trans is the actual transporting surface. So just like a shipping business, it's got a receiving where they intake, the delivery product, from the customer. So if you're wanting to ship something, you got to bring it into receiving. And the Golgi packages it up the way that a shipping service like FedEx or UPS does. And so they package it up and then they ship it out to where you want it to go. So that's the function of the Golgi apparatus in this way. Mitochondria here are always oblate. They have like this oblong shape to them. They're full of membrane crista, and these are the powerhouses. These make ATP, the mitochondria. The membrane is a double membrane on the inner and outer with a space that separates it, as you see here, so that it gives it that labyrinthine look to it. And the ATP is made through what we call the electron transport chain of the mitochondria, the electron transport chain. And so mitochondria also have the distinction of being the only organelle to have its own DNA. Make a note of that I didn't write down, but I delivered that in lecture so that you make a note. Mitochondria have their own DNA. They're able to replicate on demand. So this means that when your body needs more energy, it puts the demand on the mitochondria, hey, we need more ATP. The mitochondria sense this demand and go into a replicative mode. They begin to replicate within the cell. And so this is what explains when you begin to exercise and become aerobically fit. You first start working out, you don't want to work out, you don't have the energy. You're like, oh my God, I thought I could do 20 minutes. I can barely do 2 minutes on this bike. But you go and you build up your stamina. Pretty soon, in a couple of months, you can't hardly wait to go hit the gym and do your 20 or 30 minutes on your exercise bike or track or whatever. What is the difference? You actually have more mitochondria after having provoked them into producing ATP. So now you're like making all this ATP and you're just jonesing to go and burn it off. And this is how you get aerobically fit. Conversely, you stop working out, your body's like, well, he's not burning, she's not burning ATP. We really don't need to keep the mitochondria around. So the body begins to get rid of them. So you begin to gear down on your ATP production. So But that's true. That's the whole basis for it. Next page, lysosomes. Let me see if we have a good lysosome on this diagram. Ah, here we go. It's a little shaded, but we see it here. A lysosome are the stomach or garbage disposal of the cells. They are full of enzymes. Enzymes, of course, are molecules that are going to facilitate the metabolism of things that come into contact with them. For instance, the enzyme, my personal favorite enzyme, I don't know if I've mentioned my personal favorite enzyme on here, but I will type it in. This is my personal favorite enzyme. Alcohol dehydrogenase. Learn it, live it. Some of you use it. Maybe you didn't know what it was called, but there it is. This is an enzyme. It has a substrate. The substrate means the substance upon which this enzyme works. What do you think the substrate is for alcohol dehydrogenase? It's alcohol. Very good. Well, people are paying attention. Excellent. So lysosomes are full of enzymes. These break down substances that the cell consumes. Uh, there's multiple of them, lysosomes, I should say. There's multiples of them in any cell. If we had more of them at death, if our cells had like 10 times what they have now, there would be no body left to bury. You would just sort of dissolve into a little puddle of goo at death, because these things would then release all of their enzymes and you would just sort of self-destruct without a body. There would be no funeral or mortuary business possible because people's bodies would just disintegrate upon death. But we do have just a few, so there's always something left to bury. They're part of the decomposition process, in addition to the metabolism of materials that we eat, that our cell uses to break down for nutrition. Now, they also clean up within a cell. They also clean up within a cell. And there's a disease, a condition, this one I want you to know too, so I type it in, called Tay-Sachs disease. Tay-Sachs disease. This is a disease of the lysosomes where their metabolism is defective. The enzymes in them is defective. This is a disease that children are born with. You don't develop it during life, you're born with it. So we call it a congenital defect. That's what the term congenital means. Congenital defect. Tay-Sachs is damaged lysosomes in brain tissue. It's specific to brain tissue. So the cells of the brain, the cells of the nervous system, the lysosomes have defective enzymes. And like every other cell in the body, neurons produce trash. They produce a trash lipid in their function. Most people have normal enzymes that can break down this trash lipid and clear it out of the cell and keep the cell sweet and clean. Children who are born with Tay-Sachs, these enzymes are defective, and therefore then these trash lipids in the nerve tissue just build up. Because the brain tissue can't destroy it, it just begins to fill up each individual brain cell, And this begins to compromise the development of the nervous system so that it doesn't develop. What was born a healthy baby that looked like it could thrive and is going to be good and all that, good to go, never develops. These children are always dead by the age of 5. There's no cure for Tay-Sachs disease. Who does it affect? It affects Ashkenazi Jews almost exclusively. And I'll type that, Ashkenazi, forgive my spelling. Let's see, Jews. And so this is a particular genetic marker defect within these people that became concentrated because the peoples originated out of a part of Europe where the gene pool was very shallow, was very small for them to marry outside. And so it concentrated the lethality of its occurrence. And so now, if you have people of Ashkenazi Jewish background and they get married, they almost always have to go in for genetic counseling when they become pregnant to see what happens, because the death mortality is like 100% with Tay-Sachs. They're given counseling as to what their options are for the situation. Since again, there's no cure for this. Ribosomes are produced in the nucleoli. Remember, ribosomes are found in association with the rough endoplasmic reticulum. They are composed of what we call ribosomal RNA, ribosomal RNA. And there are two parts to the ribosomes, a large and a small subunit. So this organelle is two parts, a large and a small subunit. The cytoskeleton is composed of 3 types of material: the microfilaments, the microtubules, and the intermediate filaments. The microfilaments are composed of the protein actin, and this actin is about 5 nanometers in diameter, very small. What's a nanometer? A nanometer is 1 billionth of a meter. 1 billionth of a meter. I would like to have in the bank, in dollars, the number of nanometers in a meter stick. If I had $1 billion in the bank, I probably wouldn't be teaching. Microtubules are made of the protein tubulin. And they're a little bit bigger than 15 nanometers. Intermediate filaments, well, the protein that they're composed of depends upon the cell itself. The intermediate filaments are going to be about 10 nanometers or so. And it's easy to remember the sizes here because we're looking at microfilaments and microtubules. We got 5 and then 15. And then the intermediate one is 10, that's in the middle. So if you can go 5, 10, 15, you can easily memorize the sizes here of these proteins with their features. A centrosome is composed of a pair of centrioles. And here on the screen, we have a centrosome matrix, which would compose these 2 centrioles. So a centrosome is going to be composed of 2 centrioles. The centrioles themselves are going to be composed of microtubules. The centrioles are from where cilia and flagellum will grow from, and cilia and flagella are the motile, moving parts of a cell. They have a 9 triplet plus 2 arrangement within 9 pairs. And again, well, I'll have to find that PowerPoint because it gives a close-up and shows us this better. The centrosome will divide in the process of mitosis. It will divide in the process of mitosis, so that each new cell will get a centriole. Now, specialized features of the cell are the flagellum. Now, a flagellum is exclusive to a sperm cell. Only sperm cells have flagellum. And the only other cells in the world that contain flagella other than sperm cells are bacteria, only bacteria. We have cilia, we have microvilli. Cilia are actually the small hair-like extensions that are coming up off of a cell that will move substances over the cell surface. Microvilli are extensions of the plasma cell membrane. They're extensions of the plasma cell membrane. So we find microvilli in tissues that function for absorption, such as those tissues lining the GI tract, where you have to absorb nutrients. Vacuoles are structures within a cell. This is an organelle that's going to contain some sort of liquid. So vacuoles, most of the time, are going to contain water. Sometimes a vacuole, it will contain lipid. Sometimes a vacuole will contain lipid. And I'm looking here on our diagram to see if I can point one out to you. Now, sometimes not all the diagrams have these. They don't. It's not odd that this does not have a vacuole, but it has a peroxisome. So I'm going to get to the peroxisome here. Now, inclusions are going to be melanin and glycogen. Melanin is a pigment. It's what gives us our color. Everybody have melanin. Everybody have color. There's only one group of humans that do not have pigmentation. Anybody want to type in the chat screen what type of a human that is? Let me see.
Albino?
Albinos, very good. Albino people, albino animals have no pigmentation. Glycogen is carbohydrate. Glycogen is carbohydrate. So this is human starch, and your muscle tissues lay this up. And there is a distinction amongst humans that people that can produce glycogen and store it up and access it efficiently make better athletes. So physiologically, there's a physiological basis for athleticism in this regard. True, training and everything comes into it, but the ability to create the glycogen and then access it reliably and efficiently gives you an advantage in athletic competition. So, and in any type of event, swimming, track, field, weightlifting, anything, playing professional sports, etc. So there's something to be said about genetics and being athletic and how it does run in families, which is very true. All right, mitosis. Okay. Mitosis is cell division, and there are 5 phases, 5 phases. And I use the acronym IPMAT. to help me remember. And I'll type that in right quick here. Let's see. IPMAT is the acronym that's going to help you. This acronym will help you memorize the phases: interphase, prophase, metaphase, anaphase, telophase, and in the sequence that it goes. So learn it, commit it to memory. It will help you a lot. Now, mitosis is how your body replicates and replaces its tissue. Not all tissues of our body are mitotically active, only some, like our skin is mitotically active. The lining of the epithelium for our GI tract is mitotically active and will grow and replicate. Bone marrow is mitotically active because it's producing red blood cells that are being replaced constantly in your body. Tissues that are not mitotically active, and those were just some examples. I didn't mention hair or toenails or fingernails and stuff like that. Tissues that are not mitotically active, of course, are heart tissue, kidney tissue, neural tissue. Those are not mitotically active. They don't replace themselves. I mean, if there's a damage with those tissues, they don't replace themselves. There's only one organ in the whole body that's mitotically active and will replace itself if it's damaged, and that is the liver. Now consider, every cell in your body contains DNA. Every cell in your body contains the same DNA. Same as we see in the liver, is in the kidneys, is in neural tissue. So how come, or as I used to say when I was a child, why come it does not replicate? Because if the kidneys or heart or nerve tissue were able to replicate the way liver tissue can, nobody would ever go into liver failure. There would be no need for dialysis. Nobody would ever become paralyzed because your nerves would make reconnections and grow back. Nobody would ever die from cardiovascular disease because a damaged heart would always be able to repair itself. Why is the liver the only organ that can do this, but all the tissues contain the same DNA? Nobody knows, and they're studying that even now. So, mitosis. This is how we replace ourselves. This is how we maintain ourselves, by replicating through these phases. Now, it's interesting. Human Lifespan Project has been going on, and they have found that, right now, currently, the human lifespan stops. The longest life human is like what, 110, 118. That's about it. But they have found that genetically, humans should have a greater lifespan. Humans have a potential of not living to be just 200 or 400 or 600, but there's a potential for humans to live to be 1,000 years old. 1,000, according to our genetics, according to how we can replicate. But something has put a damper, something stops it at about 100. We just stop producing new cells. And this is part of our aging process. You don't produce any new cells. Because if you could produce new cells and continue the mitotic process, you would never physically age. So that means that if we could actually live out to our potential lifespan of 1,000 years, you wouldn't begin looking old until you hit 600, 700. You would spend up until 600 or 700 looking like you're no older than 30 years of age. 33. Isn't that amazing? Because that's kind of like the peak of human mitotic strength is at that age as an adult. So it's really fascinating. So keep an eye out for this. Now, the phases, interphase, cellular quiescence. Quiescence means that it's quietly living, it's minding its own business. There's going to be an increase in volume, an increase in chromatin, and an increase in organelle numbers in interphase. This is the phase when the cell's living its life, living its best life. The beginning of mitosis is prophase, where the cell membrane, the nuclear membrane, please make that correction, the nuclear membrane. So it's the membrane around the nucleus that will dissolve and disappear. Condensation of the chromatin into chromosomes. Chromosomes are your genes, or how you organize your genes, your genetics. Humans have 46 total chromosomes, 23 pairs, and we'll talk more about those. Separation of the centrioles, creation of the spindle apparatus. This is made of microtubules forming the aster. The nucleoli disappear, and the spindle fibers begin to attach to the centromeres. The centromeres are located on the chromosomes, and they are attaching specifically to a feature called the kinetochore of the chromosomes. Chromosomes are led around during the mitotic process. They're led around on a leash, much like you would lead around a dog on a leash. The centromere would be the collar, and this kinetochore would be the clasp by which the leash attaches. And the leash, of course, itself are going to be the microtubules. Metaphase is the elongation of the spindle apparatus and the finishing of the fiber attachment of the centromeres, movement of the chromosomes to the equatorial plane of the cell. And this is going to be waiting until anaphase, when we get separation of the centromeres. And when we separate the centromeres, the chromosomes divide and split. And then we create chromatids. So then the chromatids will move one to the other to the poles of the cell. And then telophase is the last phase of mitosis. We get a cleavage furrow becoming apparent and deepening with finally cytokinesis. So it's the appearance of the cleavage furrow first that signals telophase. And then cytokinesis is the end of telophase, when we've produced two separate cells, two daughter cells. All right, I see someone has thrown into the chart, into the chat, that the quiz is open. Yes, it is open. So at this time, I'm going to finish lecture and we're going to continue. So good luck, everybody. I'd like you now to get in and take that quiz. And again, it's not for knowledge, but it's to get you used to kind of my style of how I examine, how I test. Okay, so don't freak out about it. Don't overly stress. And I will see today's lab. You know, if you don't get there by 10:30, that's okay. You know, don't break any laws trying to get to school on time. I'll understand. All right. Okay, people. Take care, and we will see you.
Professor, where is the quiz under?
It's in module 1, and it should say quiz, practice quiz. Should say it.
Professor,
Mm-hmm.
which email would you like us to use for you? The Canvas one or the?
Either one will get to me.
Okay, perfect. Thank you.
Either one will get to me.
Professor, the lab today is for the students who have Tuesday lab, right? Because I was in lab on Monday.
No. The lab today is for people who have Wednesday lab.
Yeah, I'm sorry, Wednesday. I was in lab on Monday, so I don't.
You were in lab on Monday?
Yes.
Well, then you've already had your lab for the week.
Okay.
You only come to lab once a week.
That's what I was confirming. Thank you.
Okay. Uh-huh.
I'm trying to find the quiz and I still can't find it. Can you show us where the quiz is?
Okay, let's see.
I think it's under module 2 at the very last.
Is it module 2 or module 1? Let me see modules. Before I even share my link here, hold on. Let me see if I can find it. Modules.
It's in module 2.
Module 2. Let's see. Electric Boogaloo. Let's see.
We don't come back after the quiz, right? We're done.
No, we're done.
Okay, thank you.
Yes, your quiz is in module 2. Sorry, all. I thought I'd put it in 1. It's in 2 at the top. Lecture quiz 1, fall terminology, practice quiz. So it's a practice quiz. Yes, there we go. There you go.
So for the practice quiz, for me, it shows that's at the bottom of module 2 for the terminology, and it won't let me click on it.
Okay, does it click on it now? Let me go through and check real quick. So let's see, where am I at? Here, 0871. Let me just go through my, just give me a moment. Okay, come on, let's see. As I go through this lecture quiz, practice quiz, terminology. Yes, it would help if I launched them, wouldn't it? Let's see, practice quiz. Okay. All right, let me see. Practice quiz. Okay, launch. All right.
Professor, what time do we have until take the quiz?
Right now till 10.
Thank you.
Mm-hmm. And let me see the last one. I'm going through. Practice quiz, terminology. Okay, they should be all appearing for everybody. And it's at the top of module 2. It says practice quiz, terminology. The SP is like from spring, so it's the same from last semester.
Professor, I just checked and mine is still closed. It's not letting me click on it.
I just opened them all. Maybe
Okay.
refresh your browser.
Okay.
It should open. Is everybody able to get in?
Mine's is locked. The terminology one, right?
Yeah, it says practice quiz, terminology. Refresh
This
your
quiz is locked.
refresh your browser.
Okay, because it said it was locked February the 10th.
Yeah, mine is still locked too.
Yours is still locked? Really?
Yeah, mine's too.
I don't know about that. It should open. Let me see. Let me just check.
It's the one
something
for
real quick.
February the 10th, right?
Yeah, hold on. Well, it shouldn't say February 10th.
Mine says February 10th at 9:40.
I don't know why that is. I thought I fixed that. Okay, hold on. Hold on. No problem. Let me just fix it real quick. Okay, let me see here. Okay, what is today? The 25th. Okay. We'll have 40. Five done. Well, that's high date. Okay, high date will be and give you a little more time on it then. All right, let me see here. Fixing it. Dun, 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 dun. Okay. Dun, 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 dun. Extend the time a little bit. And then the date. Okay. All right. What am I doing to myself here? All right. 10. All right. Done. Okay. Lock date cannot be before it's saving. Is everybody into it now, or are they still hung up?
Let me open it.
Good question. After we've taken the quiz, may we leave?
Oh yeah, you didn't have to stick around. That's fine. We'll see you.
All right, bye.
Goodbye. Goodbye.
Have a good weekend.
Mine
You
still says
too.
closed.
Which lab section are you in?
Check.
I just hit save for this one.
Yeah, under module 2, it says practice quiz.
No,
terminology.
what's your lab section? Monday, Tuesday,
Wednesday. It's today.
Today's? Okay, hold on. I'm double checking that one then. All right, let me double check here. Quiz terminology. I don't know why I did that. I thought I'd set it all up and it was all ready to go. Not a problem. So let's just go and fix it. So hold on. Hold on. We're getting there. We're getting there. And then I'm extending it just a little bit here. So you have more than enough time for it.
Okay.
So that's what's taking me a little time is the extension here. Okay, let's see. And got an extra time here. Okay, saving. All right, that should be fixed. Is that fixed? Check it
Okay,
again. refresh your browser.
Okay, let me see.
So mine is not able to be open as well, but it says due date February 10. I'm not
What
sure.
lab are you in, hon?
Monday.
Yeah, me
Monday's
too.
lab.
I'm in
Monday's
Monday.
lab. Monday's lab. Okay, hold on.
Yeah.
It says February 10th.
February
All
10th.
right. Okay. Getting in there. Hold on.
Okay.
I got to do these one at a time. Apparently, that's all right. It's mass doing it for the whole class. So, okay, I see. I see.
Let's see.
Mine is also still locked.
Yeah, hold on. Are you in the Monday lab?
No, the
Yes.
Wednesday one.
Refresh your browser. I just fixed that one.
I'm in Monday.
I did the Wednesday one. Okay. 40. Okay. All right. The Monday I'm working on right now, just to make sure. Extending the time. And then the due date is the same. Okay. There we go. All right. Done. All right. So if you're on Mondays, refresh your browser and try again.
Okay.
Everybody get in?
Let's see.
Everybody get in?
Let me refresh.
Everybody get in? I'm gonna have to go. So,
Well, actually, I see mine. So I'm going to click on it now.
okay, good. So you got in. All right, good.
Yeah. Okay.
All
Spring
right.
21. Okay.
That's
perfect.
it. That's
Okay.
it.
All right.
Okay. We'll see y'all later. Bye.